Hello everyone. So I'm going to talk about food preservation, general principles, removal of microorganisms, and maintenance of anaerobic conditions. So let us see the principles of food preservation. So what is preservation? It is a process of treating and handling food in such a way to stop or slow down the spoilage and prevent foodborne illness while maintaining nutritional value, texture, and flavor. Most kinds of foods are readily decomposed by microorganisms unless special methods are used for the preservation. The chief method for food preservations are asepsis, removal of microorganisms, maintenance of anaerobic conditions, use of high temperature, use of low temperature. Drying, drying, use of chemical preservatives, irradiation, mechanical destruction of microorganisms, combination of two or more methods. Combination of two or more methods which involves any of the two or more than two methods which are mentioned above. So let us see in what way preservation of food can be accomplished. Prevention for the delay of microbial decomposition can be done by keeping out of microorganisms, which is asepsis, by the removal of microorganisms by filtration, by hindering the growth and activity of microorganisms, which can be done by low temperature, drying, etc., and also by killing the microorganisms with heat and radiation. The second one is prevention or delay of self decompositions of food, which can be done by destruction or inactivation of food enzymes by the process blanching. Prevention or delay of purely chemical reaction can be done by oxidation by with an antioxidant. Prevention of damage caused by insects, animals, and mechanical causes, etc. So let us see the concept of asepsis. Asepsis is keeping out of microorganisms as a preservative factor. The inner tissue of a healthy plants and animals are usually free from microorganisms. If there is a protective covering around the food, microbial decomposition is delayed or prevented. Examples are eggshells, skin of fruits, vegetables, etc. When the protective covering is damaged, then the decomposition will spread from the outer surface to the inner tissue, causing decomposition of microorganisms. In food industries, the prevention of food is done from the raw materials to the finished products. The food technologist is concerned with the bioburden of microorganisms. Both the number of organisms and the kind of organisms in the food is important. The kinds of microorganisms is important because they may include pathogens. The number of microorganisms is important because it leads to the more spoilage of food and becomes difficult for the preservation. The bioburden may be the result of contamination, growth of organisms, or both. This can be avoided or we can overcome this method by packing of food is widely used in application for asepsis use of carton wrapping which prevents the primary contamination during handling. In dairy industries, maintaining of asepsis done in all the steps of production and quality of milk is judged by bacterial count. In canning industries, the load of microorganisms determine the heat process necessary for the prevention. There may be heat resistant spores of microorganisms which enter through the equipment. A well sealed container would prevent the contamination after the heat treatment. So let us see how the microorganisms can be removed. Removal of microorganisms is not very efficient in food preservation. It can be done by the methods filtration, centrifugation, washing or trimming. Filtration is the only successful method for the complete removal of organisms, but its use is limited to clear liquids. Liquid is filtered through a pre-sterilized bacterial filter made up of cinder glass or diatomaceous earth or similar material. It is used for the beer, wine, water, fruit juices, etc. Centrifugation or sedimentation is not very effective because all the organisms cannot be removed. Sedimentation is used in the treatment of drinking water and centrifugation is done in milk to remove the bacterial cells. This process is called bacterifugation. Washing raw foods can be helpful in preservation, but washing cabbage before fermentation into sauerkraut and pickles removes the normal flora required for the fermentation, and washing also removes the soil organisms on the surface of the fruits and vegetables that may resist the process blanching during canning these foods. Trimming away spoiled portions of the food is important and it will be helpful in food preservation. Maintenance of anaerobic conditions. One of the preservative factors in a seal packed food may be the anaerobic conditions in the container. A complete fill, evacuation, or replacement of air by carbon dioxide or nitrogen or inert gas will bring the anaerobic condition. It is also known as the modified atmosphere. Spores of the some of the aerobic spore formers are heat resistant and may survive in the canned food, but it will not be able to germinate under anaerobic condition. Thus, anaerobic conditions prevent the food spoilage. The E maintenance of anaerobic condition.